let's talk a little bit about biochem. So when it comes to biochem, we have to understand activated carriers. Basically, understand this. Enzymes are going to catalyze a reaction. So we will see that activated carriers and enzymes go hand in hand. So let's learn them together. All right. So when we have a kinase, basically a kinase enzyme, this is catalyzing what type of reaction? Do you guys remember? A kinase reaction, basically, you're phosphorylating with a kinase reaction. Phosphorylation is correct. So remember, this is phosphorylation, but the tidbit we have to remember is that it utilizes ATP. So you're taking ATP and you are utilizing a phosphate from this ATP, changing this now to ADP. So one phosphate gets utilized. I'm going to give you an example for each. We do have fructose 6-phosphate. You'll see that this can get phosphorylated and become fructose 1,6-phosphate. Yeah, the carrier system that we have here is ATP. ATP is a carrier for phosphates. So fructose 1,6-phosphate. The difference now, we have the next enzyme, basically. On the next enzyme, you'll see it's phosphorylase. So remember that, phosphorylase. Phosphorylase is very similar to kinase. They also cause a phosphorylation reaction. What's the difference here? The difference is that it utilizes, well, phosphorylase. Think about this. Well, let's say in street vernacular, when you lace something, you spike it. You add something to it. So I see it like phosphorylase. I'm lacing this next substrate with a phosphate. So phosphorylase, you're actually putting on a phosphate. Yes, you're putting on an inorganic phosphate. So you're grabbing a phosphate from the intracellular area and you're adding this phosphate on to the substrate. You're catalyzing a reaction where you can add a phosphate. It's just like kinase, except it doesn't take the phosphate from ATP. Instead, it's taking it from an inorganic source. So in phosphorylase, for example, you have a reaction like in glycogen. I don't know if you guys remember. In glycogen, there's this enzyme, glycogen phosphorylase. Phosphorylase adds an inorganic phosphate. So now we have glucose with a phosphate group on it. So that's what it basically does in this sense. And this enzyme here is glycogen phosphorylase. So don't worry, guys. This is all review. This is all, we're going to run through it. We're going to enforce it, reinforce it. We're going to talk more about different pathways as well. This is just the beginning. We're just getting the terminology straight. All right. So kinase and phosphorylase are very similar. This one is phosphatase. Phosphatase takes away a phosphate. The name itself will help you out here. You'll see phosphatase takes it away. Okay, so it takes away a phosphate, removes it. And basically, yes, there's many reactions here that you can see with phosphatase. And one of the best ones I can say has to do with glycogen storage disease that's connected to phosphatase. You guys remember what disease that was? Von Gerk's, correct, perfect, yes. In the essence of von Gerk's, try to remember, you have glucose 6-phosphate, and you're trying to make glucose. But of course, here in von Gerk's disease, so von Gerk's disease, you're going to see it's related to the enzyme glucose 6-phosphatase. So it takes that phosphate from the 6th carbon, basically. Now you just have glucose. So when you have an issue with this enzyme, so here, let me write the enzyme name out just so it can make sense. Yes, gets trapped in the liver. And that's what causes all these different types of issues here. Perfect. All right, so let's continue. That's phosphatase, basically removes phosphate groups. Now, next one, dehydrogenase. And this, we can naturally see this in a reaction that we're going to talk a lot about, which is basically ethanol. When you're breaking down alcohol, ethanol, and you have a dehydrogenase reaction. So what is a dehydrogenase reaction? It's going to be involved with the oxidation redox, they call it, oxidation reduction. Okay, so notice this. You are now, when we talked about ATP passing on phosphates, it's an active carrier. That's what they call it, active carrier. And it's carrying and passing on phosphates. Now we're looking at this NAD and NADH reaction. So this here, basically carrying and moving along these protons. So here you have NADH. And this reaction is an alcohol dehydrogenase. Yes, adds a hydrogen. So guys, I'm going to now tell you something that can help you when it comes to remembering reduction and oxidation. So when it comes to the idea of oxidation and reduction, like we don't want to go into autopilot to think, is this a reduction reaction or oxidation? So there's a real common mnemonic out there. It's called oil rim. It's as simple as it goes. If I see that, for example, let's say BH4, if I see this coenzyme basically become BH2, I see that it's less. The number here is less. So I'm going to say that it underwent oxidation. That easy. If I see that BH2 becomes BH4, I can say it underwent reduction. The number is more. Okay, very simple, guys. So keep that in mind. So reduction versus oxidation. All right, but here you basically have the addition of hydrogen, and this is ethanol. So this is a dehydrogenase reaction. So yes, we're going to talk more about alcohol and explain exactly how it can mess up different types of pathways as well. All right. Then, of course, we have other types of reactions. A very important one is a carboxylase. So there's a reaction where you can add, in carboxylase, you're going to add a carboxyl group. Let's see how that looks. Acetyl CoA basically becoming malonyl CoA. Now, I'm not the type of guy to try to tell you to remember all these different pathways and all these different stuff, but I'm going to tell you one thing. This is a 2-carbon molecule. Yes, this reaction will add a CO2. And it needs a cofactor. Who knows the cofactor that's needed here for this reaction? This is acetyl CoA carboxylase. Biotin is correct. Biotin is correct. Yes, so you have here B7, definitely. So what's happening here in this sense? What's going on? You're going to see that acetyl CoA is now becoming a malonyl CoA, which has 3 carbons. This is important to know. This one step is important to know. Just know that acetyl CoA is growing in this reaction. Okay, I'll tell you why, because this is going to be very important in inhibiting certain reactions, and we'll talk about that as well. So far, we're just getting the names of the enzymes, and then we're going to put it into practice. So, okay, acetyl-CoA basically in fatty acid synthesis, this is the rate-limiting enzyme, acetyl-CoA carboxylase. It uses biotin basically and adds CO2. All right. We're going to see also hydroxylase. So yeah, this one is simple. This one, its own name is just telling you what it does. Hydroxylase, what does it do? Adds a hydroxyl group, right? Hydroxylase. Okay, then we have something like mutase. Mutase has the M for moving. It can grab a side chain and just move it on the same substrate, on the same molecule. It can take a side chain and move it. That's mutase. Mutase moves, all right? And a very popular mutase reaction that we need to know about is methylmalonyl CoA becoming succinyl CoA. Yes. So this is an important pathway. This is a very small pathway. They call this vomit. I know it sounds pretty bad, but valine. So this is a pathway of unwanted structures. That's the way I look at it. I say, okay, anything that could not get metabolized normally will make its way into this small little pathway. Here you have things like, let's say, valine. You have also odd chain fatty acids. Odd chain fatty acids. You'll see that fatty acids get broken down into small acetyl-CoA groups of 2 carbon. But if it's an odd chain, it's left like 3 carbons left over. It's going to have to go into this cycle that we're going to talk about right now. So odd chain fatty acids, also methionine, isoleucine, and threonine. Okay, so this is a pretty easy mnemonic. Try to remember PMS. That's it, PMS. PMS, and this S is what takes it to the Krebs cycle, the TCA cycle. So this is basically for propionyl-CoA. This M right here is for methylmalonyl-CoA. And then basically this S will be for succinyl-CoA. And this is what we find in the Krebs cycle. For propionyl-CoA, we need B7. If we need B7, then it's obvious that this is basically propionyl-CoA carboxylase. Makes sense. Important step, you use B7, right? And yeah, of course, the next step here, this is where you use B12. And this is what we're talking about. This is the important enzyme that I was just mentioning right now, methylmalonyl-CoA mutase. Okay, one last one was decarboxylase. Basically removes a CO2, right? That's basically it. Okay, good. Now we know the names. Now let me introduce to you the idea, the pathway basically, and you'll see on your first aid, when they talk about fatty acid synthesis, they don't really add too much information, which is important if you want to diagnose your disease and know it and understand it properly. So my goal here is for you to have a general understanding of the actual concept, okay? No memorization, nothing of that sort. So let's think about this. I'm going to show you an image of lipolysis. And let's start off from there. So let's say someone has consumed a lot of food. You have consumed a lot of food, you're in a fed state, you end up building up a lot of triglycerides in your adipose tissue. So this is the adipose tissue. Now time has gone by, and with time going by, you'll basically you'll get hungry. Remember, the glucose is the primary source of energy, and it lasts about 4 hours. 4 hours, you're starting to get depleted from your glucose, and now you start to activate alternative mechanisms like glycogen proteolysis. So in this sense, let's say you start to tap into lipolysis. All right. So in this lipolysis, you'll see that there's certain hormones that are going to stimulate this. So you have negative and positive stimulation. So yeah, I've seen this in a question already. Kind of tricky because decreased insulin actually stimulates this pathway. So that's like a negative feedback stimulation of lipolysis. So that was tricky because we naturally know epinephrine and cortisol are basically the main activators of this pathway. But also don't forget, decreased insulin can stimulate this pathway. All right, so let's take a look at this. What happens here? You have hormone stimulation. Hormones basically will actually bind to the surface area of the adipose tissue. Then you have activation of your second messenger system. Here in the second messenger system, you're going to see increased cyclic AMP from activation of adenylate cyclase. So you guys should practice and know your second messenger systems. It is important. There's a lot of questions just based on that. But free points if you get them. If they are just asking you about cyclic AMP and protein kinase A, if you know what they're asking, you can get the answer right. And it is high yield. You'll see that epinephrine, the main ones that we want to focus on here is epinephrine and cortisol, although there's many other like glucagon as well. Yeah, but main one basically you have here, epinephrine and cortisol. Will bind, activate protein kinase A. Protein kinase A now phosphorylates a specific enzyme here, hormone-sensitive lipase. And this hormone-sensitive lipase, now let's take a look at this and let's understand it properly, right? Let's understand it like a normal conversation. In your adipose tissue, you have triglycerides. Triglyceride is like a ball of fat, glycerol, with 3 sticks of fatty acid. So these sticks that are sticking out here, let's just pretend that's fatty acid. So that's how it's stored. Now you want to manipulate this. You want to break those little pieces off because you want to use your fatty acid. So hormone-sensitive lipase works in every step here. It works in every step. But the rate-limiting enzyme will be adipose triglyceride lipase. That is the rate-limiting enzyme, actually. That's going to initiate the breakdown, the dissociation of these fatty acids from this glycerol. And as it dissociates, it changes its name: triglyceride, diglyceride, monoglyceride, until they break down into their most simple form, glycerol and 3 fatty acids. Now, fatty acid goes out into the blood. It can't travel freely. It has to bind to albumin, and albumin basically can take it into the liver, where we will now have the process of beta oxidation. So we have to understand exactly how this works. So this first step here was just all about taking triglycerides, breaking it down, and sending them to the liver. That's the first step here. Now let me show you this other image, this other pathway basically, which is involving the actual beta oxidation. Now let's get into it. We're looking at it here. This is beta oxidation. So don't get intimidated. You'll see that your book will only have one step written here, and that's okay. You're not meant to learn all these steps, but we need to understand this mechanism. We need to understand exactly what's going on. All right, so we're at the part where fatty acid made its way inside. Fatty acid was able to enter the liver. Now, you have to activate your fatty acid in order for it to be able to enter the mitochondria. In order for it to be broken down, it needs to be activated. And that's the first enzyme that we see here, fatty acyl-CoA synthetase. So yes, this will then become fatty acyl-CoA. Now, this is a good point for us to talk about something very important. These fatty acids, they have different size. You can find these fatty acids, they can be short chain, medium chain, long chain, or very long chain. And this is important because there's a lot of different diseases that are associated with the size of this chain. So let's understand this. When we look at very long chain fatty acid, this is an issue here because we can't naturally break this down. We have something called peroxisomes. And the job of these peroxisomes, these are organelles that are found within the cytoplasm. They're supposed to grab this very long chain fatty acid and start breaking it down. And they call this action of the peroxisome beta oxidation. So it can do its own beta oxidation, but it's not a real official beta oxidation. It just makes it smaller so then it can actually go inside the mitochondria. That's what it does. It doesn't break it down to the essence of like the mitochondria does, to the smallest degree. All right. So try to understand that. Another job that the peroxisomes also do is alpha oxidation. Now, it's worth mentioning, and I'll tell you why. Alpha oxidation is just an actual process that will allow you to get rid of some kind of acid called phytanic acid. And that's it. That's what alpha oxidation does. It allows you to safely remove your phytanic acid. The reason why I'm including this into the actual, let's say, review is because if you guys have your new first aid, I'm seeing a lot of these rare diseases, Zellweger, Refsum disease. Oh, thank you. Yes, perfect, Maria. Yes, correct. Refsum disease. In real life, these are really rare, but for testing purposes, we have to know them. We have to know exactly like the pathophysiology and understand exactly what's going on. All right. So when we look at Refsum disease, the actual issue that we have here is that we cannot have any type of alpha oxidation. That'll be the problem. So you have buildup and accumulation of phytanic acid, and this becomes a huge problem. So in this case, basically, if it starts to build up, you'll start to see a lot of particular symptoms that can help us diagnose it, which is good because we can easily diagnose this. What you'll see is that it has ichthyosis. You guys remember what that was? Hyperkeratinization of the skin. Scaly skin, yes. It's like lizard skin. It's like hyperkeratinization of the skin. So yes, that's what you see with ichthyosis. That's one thing. Patients can also have cataracts. So they do have symptoms that do stand out for us to diagnose them properly. And this is a weird one. This is a weird one, but this is actually on your first aid as well. You'll see that they have shortened third and fourth toe. I mean, I don't know, people have different sizes of toes. I haven't seen any other type of issue with that one, like shortened toes. Haven't seen it anywhere. But yeah, another one that doesn't really make much sense here is anosmia. Anosmia. Basically, patient loses the sensation of smell. You lose that sensation. Okay. It could also cause polyneuropathy. So you're going to have problems with muscle weakness, atrophy, ataxia, all these other kind of issues. Okay, so how do you diagnose this for the step 2 people? Basically, you're going to find fantic acid in the plasma. It's going to actually be in the blood. Yeah, the goal is to basically reduce it as much as you can. So you have to tell the patient to eliminate anything that has fantic acid in the diet. So any kind of dairy products. That's what's huge with fantic acid, dairy products. Also beef and lamb. Basically, the treatment you'll see with a lot of these metabolic disorders is going to be a prevention of a buildup of whatever they cannot metabolize. It makes sense. All right, so that was good. That was a good deviation. Now we're going back. So getting back to peroxisomes. Peroxisomes basically are going to help us grab these. It's the breakdown in the greens those animals eat. Yeah. When it comes to peroxisomes now, we're looking at a very long chain fatty acid. It could only break it down to where there's about 8. And then once it has only 8 carbons, then it gets processed and it can enter the mitochondria and be broken down properly. So that's the peroxisome job. It does not really want to break it down all the way. So yes, you'll see this in the peroxisome. It's a very important thing that the peroxisomes can do. Also, peroxisomes also try to metabolize ethanol, and we'll see this as well. They can actually metabolize ethanol, as crazy as it sounds. They have a catalase activity. They have a catalase reaction. And you'll see this in your books as well. You'll see that it says catalase on it, and you'll realize that there's a hydrogen peroxide activity that goes on there. It's on your first aid. You guys have this information, but let me paste it so we can be on the same boat. All right. So here, take a look at this. And it should make sense. Look at this now. Catalase reaction that we see here. This peroxisome is very keen on getting rid of hydrogen peroxide, basically with this catalase reaction, and turning it into H2O. So we're already seeing 3 different mechanisms, 3 different things that peroxisomes can do. And this is a very important reaction here. So keep that in mind. Here we're seeing this whole reaction of ethanol basically breaking down into acetate. So we'll talk more about that when we finish with fatty acid. But remember as well that peroxisomes can actually eliminate hydrogen peroxide from the body. That's basically its job as well. So not only does it do alpha and beta oxidation, it also eliminates hydrogen peroxide. All right, good. So far, so good, right? Oh, all right, perfect. So we understand so far. Any questions, let me know. I can answer. Okay. So all we have spoken about was basically we started it fresh. All we have mentioned was that the fatty acid has entered into the liver. And then once the fatty acid entered the liver, fatty acid now becomes fatty acyl-CoA. Now, if it's a large chain fatty acid, not very large, we're just talking about large chain fatty acid, it needs to go in through a shuttle. And here we have a carnitine shuttle. This carnitine shuttle is very important. This carnitine shuttle will allow this large fatty acid basically to enter the mitochondria. How does it do this? It does this via the enzyme carnitine palmitoyl transferase 1, CPT1. CPT1 adds carnitine to the fatty acyl-CoA. So now you have acylcarnitine. And acylcarnitine can enter to the carnitine shuttle without any problems. So once it enters, now it needs to convert itself back into fatty acyl-CoA. Okay, and that's done with CPT2. This should say 2. So carnitine palmitoyl transferase 2. Turns it back. Good, perfect, no problem. Once it's back here, now it can undergo beta oxidation. And this is how beta oxidation looks. The mnemonic is O hot. Oxidation, dehydrogenase. We spoke about this. So now we know this. This is no problem. Whenever we see dehydrogenase, we are talking about the transfer of a hydrogen molecule. We're seeing it here with FAD, right? Is now it basically carrying FADH. And we can see now hydration. We spoke about this. Basically, you see the addition of H2O, and now you're looking at oxidation. Again, dehydrogenase. This time they're using NAD. Perfect. And then thiolysis, you break it down. So what happens? You have a huge molecule here, very huge. So let's understand this properly, because what is going on in beta oxidation? What is actually happening is that you're breaking down this fatty acyl-CoA into a acetyl-CoA. This acetyl-CoA is only 2 carbons. So if this chain was a chain of 16, now this chain is 14. And you basically have broken down, you shaved off an acetyl-CoA. You were able to shave it off. So now basically you repeat the beta oxidation over and over again until it gets lower and lower until you completely basically dissolve this fatty acid. That's what we're looking at here. Now, this process here, if the fatty acid, like we said before, if it's odd chain, if you only have 3, you can't break it down anymore. You can't break it down into any group that will make sense here. So what ends up happening is that this 3 would be a odd chain fatty acid. And it goes through the cycle of PMS. Remember that. Propionyl CoA, methyl malonyl CoA, and succinyl CoA. All right, perfect. Methyl malonyl CoA. All right, now, of course, you're going to see what does this acetyl-CoA, what is this going to do? And this is where it gets important, because this acetyl-CoA can either undergo ketogenesis or it can directly enter the TCA cycle. Yes, it can enter the TCA cycle. It will yield, it will give you 10 ATP per acetyl-CoA. Remember that, 10 ATP per acetyl-CoA. When it comes to lactic acid, we net 2 ATP. When it comes to a molecule of glucose, we're looking at 32 ATP. Okay? So yeah, here we're looking at this. This can enter the TCA cycle or it can undergo ketogenesis. So yes, we understand that. That's good. Now, let's understand what happens in the sense of synthesis of fatty acid. And this is where I would recommend you open up your first aid book, because naturally you'll see that a lot of the information of this pathway is just left empty. It's just completely like they just didn't really feel like putting in more materials and adding in enzymes to this list. So they kind of left it empty. So I'm going to screenshot exactly what I'm talking about, and we're going to fill it up with the enzyme names. And all right. So acetyl-CoA carboxylase. This here will be the rate limiting enzyme of this pathway of synthesis of fatty acid. So it should make sense. If you have a huge buildup of citrate, if citrate starts to build up, basically you're at a fed state, your substrates from your TCA cycle are building up and accumulating, citrate accumulates. So you're going to see that citrate, whenever it's high, it incentivizes the actual synthesis of fatty acid. Basically, it will be able to leave through the citrate shuttle. So remember, carnitine is for breakdown. Imagine a car that's breaking down. Citrate is for synthesis. So it makes its way up and it breaks down here with citrate lyase into acetyl-CoA. And remember what we said, acetyl-CoA grows into malonyl-CoA with acetyl-CoA carboxylase. And yes, this here is stimulated by insulin. And this is going to allow fatty acid synthesis. So we need to understand that. because there's a very important pathophysiology that we're going to see. We have to understand basically hepatic steatosis, non-alcoholic fatty liver disease. How does this happen? This is all within this pathway that we're talking about. If we look at hepatic liver disease, let's understand that. If we look at hepatic liver disease, there can be an alcoholic type. When someone is consuming alcohol, what is happening exactly? You are causing a disbalance with your NAD and your NADH. We agree on that, right? Because your alcohol dehydrogenase is forming higher levels of NADH. That makes sense. So this here will cause havoc. If you have more NADH and lower amount of NAD, you are unbalanced. And your body tries to help you by doing several things. So you have several ways to try to compensate, to try to get it back into the correct balance. And naturally, the way that we all learn, I'm sure you learned it this way. You look at pyruvate turning into lactate. We understand that one, right? That's the one that everyone understands, because on this step here, what you end up seeing is that basically your NADH becomes NAD, right? And that is a nice one to understand, but it gets more complex than this. That's just one thing that happened to pyruvate. And this causes basically, yes, this will cause lactic acidosis. We know that. So lactic acidosis, that's good. Now let's learn all of them. Let's be complete here. Another one basically that also higher content of calories, 7, you're not able to break down all the calories, the excess will deposit in the liver. Yes, and this is how it deposits, not only the deposits, but it also shuts down the other pathways. So check this out. You'll see that oxaloacetate, instead of it moving forward in the TCA cycle into citrate, instead of it advancing, guess what it does? It actually ends up going back to malate. So check this out. This is very interesting because now oxaloacetate, it's only doing this because this reaction here actually tries to help you. This reaction will take an NADH and turn it into NAD. So here we have so far 2 of them, 2 reactions that we're looking at. And we said the consequence of the lactic acid building up is a lactic acidosis. Okay, that makes sense. Yes, basically here, you're shutting down your TCA cycle. So here you shut down your TCA cycle. So your TCA is off. You cannot produce ATP. Electron transport chain will not see any kind of NADH or anything of that sort. Now you're having this problem. So could you explain again? Yes, definitely. So on the TCA cycle, you'll see in the TCA cycle, we have oxaloacetate, coming citrate, isocitrate, alpha ketoglutarate, right? Basically, this is what's naturally happening in the TCA cycle. But when you have a disbalance, when you have way too much NADH in comparison to NAD, your oxaloacetate tries to help you. Yes, the lactic acidosis we understand, and that's great because that's one of the biggest problems. Lactic acidosis, definitely. You'll see a lot of manifestation of that. But here's another reaction that people really generally don't talk about. And that's what we want to do today. We want to understand these things, these small little things that basically cause these other types of diseases. So definitely here, you'll have a patient who cannot run the TCA cycle. And that's the issue here. So oxaloacetate, instead of it going forward to citrate, guess what it does? It betrays us. It goes backwards to malate. Why does it do this? Because this reaction here catalyzes a reaction in where basically we take NADH and we make it NAD. So it's trying to help us. It's trying to get the balance back. All right, so that's 2 reactions so far I gave you. 2 reactions. All right, another reaction you're going to see. So that one you can see, that's a TCA reaction. And this one is interesting, but you will see that ethanol, basically. So, okay, another reaction, basically, if you have continued to drink alcohol and it kept on happening, of course, we have a buildup of acetate, right? Acetate. We understand that. Natural buildup of acetate. Why? Because, of course, ethanol breakdown. What people don't see is that this acetate can become acetyl-CoA. In your book, they end it with acetate. They don't continue the line. Look at this. It ends here. But that's not the real story. In the real story, it continues and becomes acetyl-CoA. And that is very important. And I'll tell you why. That's very important. So far, we mentioned three things. We mentioned anaerobic glycolysis, which is basically buildup of lactic acid. We've mentioned increasing the levels of acetyl-CoA. And we mentioned as well also acetate, turning off the TCA cycle. Now, if you have a high level of acetyl-CoA, guess what happens? Exactly what we were mentioning before. We mentioned the whole synthesis. Where is it at? We were mentioning the synthesis right here. And this whole synthesis is based on high levels of acetyl-CoA. So we are provoking the synthesis of fatty acid by creating a lot of acetyl-CoA. Notice that. If the patient is intoxicated with alcohol, they are creating an incentive for the fatty acid synthesis pathway to occur. So now guess what you have? You have a buildup of fatty acid. We have these evils are now happening all at the same time. We have lactic acidosis. We have basically the inhibition of TCA cycle. All this building up just because of too much alcohol in the system. Okay. Now, so what is the problem here? What happens? There's one more thing that we need to talk about as well. And this is something that, of course, it's not really found in many books, but you'll see it if you look for it. DHAP. You know, if you guys remember DHAP, this is part of the glycolysis. Shunting everything to create fat and reduce glucose production, so it can lead to hypoglycemia. Yes, these patients will have hypoglycemia. They cannot run the TCA cycle. And yes, not only are they increasing fatty acids, we're going to see right now this DHAP, and you'll see DHAP on the glycolysis. DHAP is going to basically become glycerol. Glycerol 3-phosphate. And guess what, guys? Guess what? Guys and girls, guess what? Remember in the beginning, we were talking about triglycerides? We said that a glycerol with 3 fatty acids. 3 fatty acids. That's triglyceride. And this right here is the pathophysiology of alcoholic hepatic steatosis. This is how it happens. Basically, this starts to accumulate in the cytoplasm of the liver, and it increases, gets very high. And it's affecting the cytoplasm because acetyl-CoA is outside in the cytoplasm at this moment. So guess what happens? When we look at the liver, let's say here we have mitochondria. We have mitochondria. Mitochondria has AST in the mitochondria, and it also has AST in the cytoplasm. But we have an abundance of ALT in the cytoplasm. So if you look at your LFTs, your liver functioning enzymes, you're going to see that you're going to have a increase, more ALT than AST. So that ratio will be increased because the liver is getting infiltrated with a whole bunch of fat. Yes, ALT, so lipids. That's how you can remember lipids. That's one way of remembering it. So that's the pathophysiology of it. That's how these patients have this type of issue and these problems. So what else? Also, there's going to be stages to this because at first it's just a simple accumulation. So yeah, let me see here I have on my notes. And I'll send you guys the notes that I have. I was able to write the different stages which you'll see in these problems. So hepatic steatosis is the first stage. Steatosis. And this basically is going to be is the most benign one, and it's just basically the first stage. And this is filled with triglyceride deposits. Triglyceride deposits. Now, this continuously gets worse and worse. Okay, sure. All right. So yeah, that is a good question, Alejandra. I'm going to show you. So when it comes to certain types of organs in our body, we have two things that we like to measure: functionality and damage. Those things are not synonymous. They are actually different. And you'll see, for example, with the kidney, how do I measure functionality? I measure it with the GFR. Basically, I do an equation with creatinine, and I can find out what the GFR is. And I can measure that to know the functionality of the kidney, for example. If I want to measure damage, I measure proteinuria. Proteinuria. Albuminuria if I want to be more specific. So albuminuria if I want to be more specific. All right, that's the difference here. One of them is functionality, and the other one is basically damage. And we start to see this same concept in a lot of different tissues. So, for example, in the muscle, whenever you have damage, let's say, of muscle tissue, then a lot of enzymes would be a lot more permeable. So you have that creatine kinase that can leak out. And definitely, if it starts to leak out, we can call this myositis. If you have myoglobin leaking out, they like to call this rhabdo. And this speaks to damage. Damage. Okay, functionality is done at a more clinical level with the patient is contracting his muscle. Either he has weakness, spasticity, or anything different like that. That's muscles. Now let's look at the liver. That's what I was talking about right now, liver. Liver has functionality and it has damage. So if we're going to understand the actual functionality, I'm going to tell you guys. You guys probably had a question on this, especially if you guys have done UWorld. TT, thrombin time. Thrombin time. I saw a question where they actually made it PTT. But thrombin time. Remember that our liver is responsible for making antithrombin 3. This speaks to the functionality. If there's a problem with thrombin time, then our functionality is taking a hit and the prognosis is really bad. Because if your liver is not functioning, that's not a good sign. So function, thrombin time. Now let's look at damage. Damage is what I was talking about right now. What I mentioned was liver enzymes. So yes, you have GGT, you have your AST, and you have ALT. So let's understand this. In the hepatocytes, you'll have mitochondria, and the mitochondria, within the mitochondria, you have AST. But you also have AST in the cytoplasm. But what you have actually ALT at a very high level in the cytoplasm and almost no ALT in the mitochondria. So if I have accumulation of glycerol and I have synthesis of fatty acid, I'm going to have triglycerides that are now going to occupy the cytoplasm. And these triglycerides are kind of big. They're not small. So they start to occupy too much space within this hepatocyte and they can start causing damage. What type of damage? Usually it starts off with reversible type of damage, swelling of the cell. That will lead to permeability. That would lead to enzymes that can leak out of the hepatocyte. So we will have leaking out of AST and ALT, but ALT is more abundant at a cytoplasmic level. So that's what you'll see here, increased ALT. because this is more of like the minor. This reminds me of the creatine kinase. Because why? Because it's something where if your muscle is damaged, you'll leak CK, creatine kinase. But if your muscle is getting destroyed, then you leak myoglobin. So it's like this is the more benign one at this point, more the cytoplasmic one, right? Makes sense. So that's why we see that ratio, ALT is higher than AST. So yeah, these are basic concepts, and I feel like they really should teach this in med school in a point where everyone can understand it, because everything, for example, in step 2, when you start to look at labs, you'll start to understand like what's going on. If you see a patient who has very low GFR, this is how they actually grade chronic kidney disease. They actually grade it by looking at the functionality and the damage. They can say, oh, he's grade 3A, for example. If they measure GFR more than 60, but proteinuria gets really bad, then they modify the score. So yeah, this is useful in clinics. Yeah. Thank you. Definitely. This is useful in clinics as well. All right. So basically we mentioned, this is how it happens. You'll have, as it gets worse, of course, you'll have steatohepatitis, and this is when you have ballooning of those cells. High AST-ALT ratio means that beta oxidation is higher, trying to process all the excess fat. But when ALT and AST is higher, you produce fat surrounding the liver, since damage is more in the surface and less in the mitochondria. In this specific case, yes. Why? Because the buildup of triglycerides that you're getting, it's happening in the cytoplasmic level. This is acetyl-CoA. Oh, let's see, acetyl-CoA. It's building up because of that ethanol turning into acetate. And this acetate is basically accumulating a ton of acetyl-CoA on the cytoplasm. And that's what's causing that fatty acid buildup. And then also what I mentioned last. So let's not forget what I mentioned last. I mentioned DHAP. There was something called DHAP. DHAP is turning into glycerol 3-phosphate. Glycerol 3-phosphate. So think about that. Now you're having fatty acid and glycerol, triglycerides basically. So that's the pathophysiology of it. That's how we understand it. That's what happens. Okay. All right. So, we were at the point where we were able to have acetyl-CoA in the liver made by beta oxidation. So beta oxidation happened, and now we have acetyl-CoA. And I mentioned to you that acetyl-CoA can either undergo to the TCA cycle, or it can go and make ketone bodies. That's what I told you. All right, let's see. So if we look at it like this, we have a ton of acetyl-CoA. What happens here? Yeah, thank you. When we have acetyl-CoA, you're going to notice that this acetyl-CoA undergoes a process where there's it condenses with another molecule of acetyl-CoA, because you're making so much acetyl-CoAs here from beta oxidation. So this acetyl-CoA joins up with this other acetyl-CoA, and now you have 2 acetyl-CoAs. So now there's 2 acetyl-CoAs that are hanging out together here. Now, in this process of ketogenesis, the next step will be an enzyme basically called thiolase. So what we're seeing here basically is more acetyl-CoAs are joining together. So when you have 2 molecules of acetyl-CoA, they condense together, they make something called acetyl-acetyl-CoA. These 2 molecules has joined together to make one molecule called acetyl-acetyl-CoA. And now guess what happens? It's crazy. Believe it or not, another acetyl-CoA joins the mix. And now you end up having HMG-CoA. So yeah, if you look at the actual enzymes here, you'll see that this here is thiolase. You'll see that this here is HMG synthase. All right, so the final step here in ketogenesis is that this HMG-CoA will basically, you'll have a HMG enzyme here is called, very important enzyme. This is the process of the fates of pyruvate decahyl cycle. No, but that's another thing I wanted to talk about today. Time is flying. We could actually mention that actually. Yes, but that's protein breakdown. That's different. Here I'm just ending the whole ketogenesis, mentioning the process of ketogenesis. So HMG-CoA is basically here, we're seeing that we just keep on building up acetyl-CoA. Think about that. Ketogenesis is just a process where I'm taking acetyl-CoA, adding another acetyl-CoA, and further down the line, I add another one. And now I have HMG-CoA. And this HMG-CoA, basically, you have an enzyme here called HMG-CoA lyase. And this lyase is going to create our acetoacetate. This is what we are looking at when we're mentioning the ketogenesis, right? This is the whole idea of ketogenesis. If this gets reduced, if this acetoacetate gets reduced, then we have beta hydroxybutyrate. That's another type of ketone body, right? So here we have beta hydroxybutyrate. Boom. These two ketone bodies, perfect. And they go out and travel within the blood. They go out and travel within the blood. Now, there's something that can happen to acetoacetate. In the blood, it can actually have a decarboxylation. And when it gets when there's a decarboxylation reaction, it can actually turn into something called acetone. The actual lungs can breathe this out. And that's what gives that fruit. They describe it as a fruity smell, but to me, acetone does not smell like fruits. It kind of smells like nail polish remover. So yeah, but you'll see in questions for question purposes, you'll see that they describe it as a fruity odor. And this gets eliminated by the respiratory tract. So this can happen when it gets randomly decarboxylation, spontaneous decarboxylation occurs, all right? Okay, so what happens? This basic, these kind of ketone bodies make their way into the muscle and they get used. You'll see the TCA cycle. This ketone body, I'm just going to put a K, will react succinyl-CoA on the TCA cycle. And now it'll break itself back down into acetyl-CoA. So it's all about packing up the acetyl-CoAs and then unpacking them. That's it. That's it. And this turns into two molecules of acetyl-CoA. So now you have acetyl-CoA and acetyl-CoA. Two of them. And then this can enter into the TCA cycle. And that's it. That's it. So yes, you're going to see something. Yes, that is true. In DKA, you are, remember what we said basically in DKA, these patients, a lot of type 1 diabetic patients are dependent on insulin. They are dependent on insulin. Their insulin levels are very low. And in the very beginning of this class, we mentioned the stimulation of lipolysis. We said that a very low level of insulin can stimulate lipolysis. So that will initiate this whole fatty acid breakdown, and that will provoke ketogenesis because the patient needs a source of energy. So yes, this is what's happening here in that sense. Hopefully you guys understand what I'm saying. This is just ketogenesis, basically simplified. I know it looks kind of crazy. I have a better drawing. I can send it to you guys. This is what I'll do is I'll send you the pathways that we're talking about. All right, so let's see. I started at 1. I'm going to give one more pathway. We have time for one more pathway. Yes, correct. Low insulin will trigger hormone-sensitive lipase, and that's a negative feedback. Positive feedback is glucagon. Positive feedback is basically epinephrine is the major one. Epinephrine and cortisol. Cortisol has the effect that it remodels, though. It can actually mobilize fats. And that's what you see with patients who have Cushing's. they can have central obesity. So these hormones that are stimulating in stress, they kind of have a difference between them. For example, glucagon is going to be stimulated in a stressful metabolic situation where you lack energy. Now, norepinephrine and epinephrine get activated when you're in a fight or flight response. Your body doesn't know if you might need all the energy in the world to try to survive. And cortisol gets activated at a more chronic level. If you basically are needing, let's say, some kind of energy source for a longer period of time. And growth hormone gets activated when you go to sleep. So they all these things have a different type of little edge to them. Yes. Thank you, Kim. Yes, definitely. Make sure you guys follow Instagram. I'm going to drop it here. Yeah. Definitely. Guys can contact me if anything. Let me continue with this last pathway I'm going to talk to you about. I know it's out of the blue. We mentioned everything that has to do with fat. We mentioned ketone. So we mentioned ketogenesis. We mentioned the breakdown of ketone bodies. Yeah, if I would do another class, what I would do is now just bombard the whole lecture that we just did with pathological association. So we'll talk about diseases. Like that'll be good to put a layer on it. For example, say, okay, well, what happens if we could stem off the DKA, for example, the management of DKA and all that. That would have been good. But yeah, I got like a couple minutes left. I do have to go. I have a session later on. So ketone bodies, we mentioned lipolysis. Damn, my spelling now is just really bad. I'm hypoglycemic at the moment. My glucagon is active. My epinephrine is going up. So lipolysis, I know I'm spelling it wrong. And we also mentioned, beta oxidation. And we mentioned fatty acid synthesis. And we mentioned enzyme terminology. I think that's awesome. We did that like in an hour and 20 minutes. But what I would have liked to do is add a whole bunch of other normal first. Yes, add layers to it. Dr. Lopez, you know what I'm saying? Like a drill, just throw it at him. Like, okay, what's this disease? This disease, differentiate. Yeah. So last pathway that I am going to include as well. Let's see if we have time for it. Very short. I'm just going to breeze through it. All right. This here is the urea cycle. And this is a simple concept. It's the whole basics of biochem. There's a certain rule here. So think about it like this. If you have something like a substrate, basically, that cannot go on to the next level, cannot have any enzyme catalyzing a reaction, then this substrate will build up and your product will be low. That's a very important biochem rule. Learn this rule, because this rule here, you're going to see that this can be manipulated. This rule also includes the fact that if I have increased product, I decrease the incentive to make more. And this is where I see a lot of people fail on questions, because they'll ask about a certain type of substrate that will inhibit glycolysis, for example. Like if you have certain buildup of a certain substrate, that's like in the TCA cycle. If you have, for example, the best example is ATP. If I have increased ATP, I am basically inhibiting the breakdown of glucose because I have product. If I have a lot of product, I inhibit the incentive to make more. Like in sphingolipidosis, decrease the incentive to make more, definitely. AMP is the opposite. If I have adenomonophosphate, then I need more ATP. I'm running low. So that's an important concept when it comes to biochemistry, because you're going to see that this can be manipulated. I can stop a whole pathway by giving a product. Okay, but when we're looking at urea cycle, this here has to do with protein metabolism. Let's remember, that protein amino acids have this ammonia. They have this nitrogenous group within its structure. And this nitrogenous group, you have to get rid of it. If you do not get rid of it, it can cause damage in the body. So basically, amino acids, they get processed through this. So let's understand this. I have an amino acid. Basically, proteins can be utilized as functional or structural types of protein. Not only functional and structural, you could also use protein as a source of energy. So a protein can be utilized to build, for example, collagen. A protein can get utilized to catalyze like an enzyme, for example, and can do a reaction. Or it can be used as a source of energy. So here we're seeing an example where this amino acid is basically getting converted into alpha keto acid. Now, what is an alpha keto acid, you might ask? What is that? Don't get confused. This is something very simple. It can be alpha ketoglutarate. It can be pyruvate, right? It's okay. It's a list. It's a whole bunch of different substrates that have this alpha keto acid group in it. So pyruvate, alpha ketoglutarate are two of them that we should know. So what we're seeing here is the passing on. It's like hot potato. They're passing on this ammonia from one amino acid on to another amino acid. Now we have glutamate that grabs it. Here you have alanine that grabs it. So let's understand here. An important point we should make is that this glutamate and this alanine, they can both travel in the blood with ammonia to go to the liver and run the urea cycle. It's not only alanine. In your books in the first aid, you'll see it drawn off like alanine, but it's two things. Glutamate and alanine can both travel through the bloodstream in order to reach the liver to run the urea cycle. The purpose of the urea cycle is basically to take this ammonia and convert it into urea. Urea can get excreted through the kidneys without any problem. So it's detoxifying ammonia, basically. That's the whole process when it comes to this idea. The whole mission of the urea cycle is to break a amino acid down, take the ammonia group, convert it into urea, and then excrete it through the kidneys. That's it. So we're seeing it here, this process that's passing it on from one amino acid to another. And during this process, you are catalyzing several reactions. Basically, you're seeing pyruvate turning into alanine. Pay close attention to that, and I'll tell you why. Because we mentioned before, when you have an unbalance in NADH versus NAD, your pyruvate will be incentivized to turn into lactate. Your pyruvate is incentivized to turn into lactate. So your pyruvate will not be available for this step. So look at this. This is intense. Here we're seeing that lactic acidosis can cause hyperammonemia. So you can have high levels of ammonia just because you have lactic acidosis. So if you ever wanted to connect those two things, now you're seeing why. You need pyruvate in order to convert it into alanine. So alanine can carry the ammonia group. If all your pyruvate is busy turning into lactate, you cannot run this cycle. You can't run this pathway. And then you'll have accumulation of ammonia. So this can cause hyperammonia. All right. And yeah, so this is one of the main issues here causing hyperammonia. And you're also going to see you have protein catabolism. Involves transamination reaction, passing on the amine group from one amino acid to alpha ketoglutarate. Okay, hyperammonemia incentivizes this step at an excessive rate. Yes, that's another thing. You can also have inhibition of the TCA cycle in hyperammonemia. So let's understand this. Remember all these crazy type of symptoms that these patients have, the asterixes when their hands are shaking. They're also found in low energy state. These patients are also found in a low energy state. Why is this? It's because you're depleting the alpha ketoglutarate at a level of hyperammonemia. And let me explain that so it can make sense. If I have an issue with processing the ammonia, let's say there's an issue processing this ammonia, the actual, let's say, body itself will try to run this pathway regardless. It will try to let's take the alpha ketoglutarate and turn it into glutamate. So yeah, but it does this passing on this group right here. When you're doing this, you start to deplete your alpha ketoglutarate. So this can cause the inhibition of the TCA cycle. Alpha ketoglutarate is utilized in the TCA cycle. Yes. Yes, definitely. Yep. And that's basically causing this problem where you have a low energy state. Patients are now at low energy state. And that's what you'll see because all that alpha ketoglutarate was turning into glutamate in hopes to pass this ammonia group off. Yes, the GABA effect happens when the brain now tries to help you. The brain now tries to take up that ammonia. It tries to basically change this into glutamate. And what ends up happening is that if you guys remember in neural, well, one of my favorite subjects is neural. In neurology, you're presynaptic. You have excitatory neurotransmitters and inhibitory neurotransmitters. So an excitatory neurotransmitter is glutamate. Glutamate. It'll be released, or mine, I think mine. It gets released. It builds up and it gets released. And that's what causes that asterixis. So in ammonia, you'll see that basically this will get you'll have a process where your astrocytes can take up the ammonia, trying to help you, trying to save you. And what ends up happening is that this ammonia, at the end, you have conversion of more promotion of glutamine. And in the end, glutamine will serve as a actual stimulator, as a neurotransmitter that will actually stimulate. And that's how they get asterixis. That's how they have their shaky hands. So yeah, remember that. And yes, you are right too. When it comes to the GABA effect, that's already death. When we're talking about acidosis, a patient with acidosis at a high level, towards the end of it all, remember we did neuro briefly. We had the brainstem. The brainstem had the midbrain, the pons, and the medulla. You have something called the medullary pontine respiratory center. This center here allows you to have respiration that's automatic. It's automatic and it's driven by a lot of chemo type of receptors. So whenever you're acidotic, your respiratory rate increases without you even thinking about it. That's because of the medullary pontine respiratory center. Now, if you have a high level of GABA, if you have lactic acidosis for a long period of time, a lot of things start to change in your body, a lot. One of the things are phosphate becomes phosphoric acid, glutamate becomes glutamic acid. As this continues and it goes on, you'll see, yeah, there's a lot of different things that happen. It's crazy. It's a big subject there when you talk about lactic acidosis. You have albumin that releases the calcium that it has. It releases it and it replaces that calcium with proton. Besides lactose to reduce ammonia, is there another natural way of doing it? Rifaximin works very similar to rifampin, but it does not get absorbed. It stays in your GI tract. And in the GI tract, it will basically kill off the bacteria that's responsible for promoting that conversion of ammonia. So you have a conversion of ammonia that happens on your GI tract, and you could actually decrease the conversion of ammonia by giving rifaximin. Yes, rifaximin. Let's continue with urea cycles. So yes, okay, now we were up to this part of the story where alanine traveled through the bloodstream to make its way into the liver. Once it makes its way into the liver, something very important that you guys need to remember, alanine now is able to become pyruvate again. So you're not really wasting anything here. You see what I mean? It's able to, from pyruvate becomes alanine, from alanine becomes pyruvate. And in this sense here, this one reaction is able to pass on this ammonia group to glutamate. And this is the very important part because glutamate is the one responsible. Glutamate is the one responsible for isolation of this ammonia. So finally, that hot potato game is over. They passed on that ammonia like a hot potato. Nobody wanted to stay with that ammonia. They did not want that in their hands. And this enzyme was responsible for releasing that ammonia. So this enzyme that you see here, glutamate dehydrogenase, very important enzyme. Yes, it was a hot potato. Nobody wanted that. That was like no good. They grabbed it and just threw it on. The last one that stayed with it was glutamate. And glutamate said, okay, I don't want it either. And they let it go with glutamate dehydrogenase. So this high yield enzyme right here, because this is the one that is responsible for isolating that ammonia. So you have to isolate it. You have to have it free in the liver. Now, why do we have it free in the liver? Because now it's ready to undergo and the urea cycle. So yeah, let's take a look at that. Now that it's isolated here all on its own, it's able to join up with bicarb and 2 ATP. Bicarb and 2 ATP. And it's able to now enter the mitochondria and basically become carbamoyl phosphate. Carbamoyl phosphate. Now, it's something very important. I should tell you that carbamoyl phosphate is also found in the cytoplasm. And it's part of a substrate of the de novo cycle when you're making these nitrogenous bases, when you're making your thymidine, you're making your uridine, cytosine. It's found in the cytoplasm, in the de novo cycle. So carbamoyl phosphate is 2 areas. You're seeing here it's in the mitochondria. Don't forget, you could also find it out there. So the enzyme responsible for the reaction here that we see, the actual synthesis of carbamoyl phosphate, is carbamoyl phosphate synthetase 1. That will catalyze this reaction. And it's controlled by something called acetylglutamate. So how do you remember this? Remember, glutamate was the guy that let it go. It's like that guy that just keeps coming back. He let it go, but again, he's coming back. He wants to take care of it somehow. He's there stimulating this enzyme. That is the allosteric stimulator. It is required actually for activation. It'll actually activate this enzyme. And yeah, so it says here, it binds to a non-active site on the enzyme to increase the enzyme activity. And you need to have it here. This has glutamate in its name. Don't forget that. And that's what stimulates this enzyme. That's the rate limiting enzyme for this urea cycle. So what ends up happening here is that carbamoyl phosphate now is able to enter this urea cycle. So I have a mnemonic here for you guys. You guys can remember this. Guys and girls, the mnemonic I have for TCA cycle, I don't know if Lopez shares his mnemonic of old criminals in Kansas sell sex for money. I don't know if that's the one you use, Lopez, but here I tweaked it a little bit to old criminals are Argentinian. And nothing against Argentinian people, but yeah, just the Argentinian, it's like arginine. It's like right there, you know, like you don't have to modify it too much. And then check this out. This is the good part about it. After criminals, you say AFU. And that's important. AFU. It's important because these are the actual substrates that are involved in joining the cycle or leaving the cycle. So aspartate, after the C, after citrulline, aspartate will join. So after criminals, you say AFU, and you'll see that aspartate joins, fumarate leaves on the next step of arginal succinate, and then urea leaves on the following step. So there you go. Here you have your mnemonic for this cycle, and you also have the substrates that are entering and leaving. The first one is entering, the other 2 are leaving. Yeah, definitely. Yeah, man. Yeah, Argentinians, they're doing it in soccer, man. They're doing it. Carbamoyl phosphate, basically ornithine transcarbamylase. And we're going to talk about differentiating these problems. They have uric acid or auric aciduria. I've seen so many students get these questions wrong. We're going to organize them so you can get it right. Right? We don't want to get these wrong anymore. So let's understand this. Carbamophosphate. Here we say it joins with ornithine and becomes citrulline because of the OTC enzyme. Then, of course, you see this next step. Citrulline becomes arginosuccinate from arginosuccinate synthase. So urea itself has two molecules. It has two. I didn't take the chemical composition of it, but you'll see two ammonias. Urea itself has two ammonias. You're probably thinking, well, where's the two ammonias coming from? One of them is from this aspartate right here. This is where aspartate gets metabolized. The other one comes from that basic group that we formed, this nitrogenous group from here. So we're seeing this nitrogenous group right here, and we're seeing aspartate right here. So basically urea is made up of those two. So here we're seeing that arginosuccinate turns into arginine by arginosuccinate lyase, and then arginine basically gets converted into ornithine by the enzyme arginase. Perfect. We understand this. So how do we get the answers right? What's going on here? What's the connection in all these kind of pathologies? What's going on? Let's make sense of this all. Remember I mentioned to everyone that there's another carbamophosphate that's in the cytoplasm. This is the carbamophosphate that I was referring to. It's again, you see glutamate here again. He's always there. This glutamate basically is with CO2. And notice this is you have to use your brain to try to see contrast. CO2 is like an acidic molecule. And here you're seeing that this here bicarb is an alkaline molecule. So I start to draw differences in my brain right away, just so I can remember this. In this test, you just have to use everything you got to remember everything. So much information. So I'm seeing here a contrast. I'm seeing that CO2 at the cytoplasmic level, but in the mitochondrial level, it's bicarb. Okay, that makes sense. Does one take over when the other one isn't working? If so, when does it happen? No, I'll explain. So both of these are they're not related to each other. One of them, it's made inside the mitochondria. The other one, it's made they never even see each other. Let's put it like that. They never even see each other. This carbamophosphate gets made in the mitochondria. And the mission of this pathway is basically to make thymidine, to make uridine, and to make cytosine. That's the mission there. Okay, perfect. But there's a problem. And we're going to mention this, and let me write all over it so it can make sense. Okay, so I'm going to screenshot this and we'll put it on the board. And let's talk about this. Here comes the problem. If you are deficient in this enzyme right here, UMP synthase, if you are deficient in this enzyme, then you're going to build up higher levels of boric acid, right? They call this boric aciduria. Now, for some odd reason, they don't want you to know the enzyme. It's not written anywhere. They don't only written in a question, for example. They'll put it up in a question for you, and it'll be hard for you to connect because you only learned it as auric aciduria. So tether this enzyme to this condition, UMP synthase. When you have inhibition, when you have a problem, CP1 is messed up, so you shunt to the other CP2. So hold on. For this one, pretty much CP1 is messed up. Not on this one. CP1 on this disease, CP1 is working. CP1 is working. Everything's good in CP1. This isn't, it's actually, you're going to see that they draw a big opposite here. So yeah, I'm going to go through it. Let's go through it and let's understand. Here, you're going to see that carbamophosphate here, for example, let's understand. Here, you're going to have at this level, there's 3 different diseases we have to differentiate for. UMP synthase deficiency is what we find in auric aciduria. We also have another disease. So I'm going to present them first, then we're going to differentiate them. We can understand it properly. CPS1 deficiency. So if you have deficiency of this enzyme right here, this is a whole nother disease, basically, CPS1 deficiency. The last deficiency here that we have to learn how to differentiate is OTC. So these are the three enzymes. OTC was the enzyme responsible for turning ornithine into citrulline. And this is inside the mitochondria. So let's look at that. One enzyme is the rate limiting enzyme, the one that creates carbamoyl phosphate inside the mitochondria. So don't worry, guys. I'm going to explain it and we'll re-explain it. You guys can ask me any questions as well. The other one was the enzyme responsible for furthering along the metabolism, the cycle of the urea cycle, basically, the next one. So those are the three different ones that we have an issue with. Now, let's understand them. Let's look at it. Let's look at it with the entire picture here. When we look at the urea cycle, you're going to see this. Hyperammonemia is going to be found in these two enzymes. Why are we going to see it in these two enzymes? I'll tell you why. These are enzymes of the urea cycle. These are enzymes of the urea cycle. The enzyme I presented to you here, the UMP synthase, is not an enzyme of the urea cycle. It's an enzyme of the de novo synthesis. Okay? So let's put this into our mind. When we look at hyperammonemia, right away, hyperammonemia should let you know right away that this is a problem of the urea cycle. All urea cycle conditions will have hyperammonemia. There's only one that could have it normal. And this is where you can get a little confused. So let's just put a side note on this one. Normally they all have hyperammonemia, but there's one. Of course, it has to be that one, right? For some reason, it's this one here, the last one, arginase. And I'll tell you how to get the diagnosis here for all of them. All you have to do is follow the rule that we mentioned, the biochemistry rule. If you lack arginase, arginine will be high, ornithine will be low. It's going to follow that same exact pattern with all the enzyme deficiencies here in the urea cycle. They follow that pattern, which is great. For example, if you have arginosuccinate lyase deficiency, arginosuccinate is going to be high. Arginine will be low. It follows the rule. Okay, it follows that rule. Now, what I was telling you was that all of them have hyperammonemia because you're not able to basically run the urea cycle, except for arginase. For some reason, this one is the only one that can have mild or no hyperammonemia. So be careful with that. Be careful. All others will have it. Okay, so like I was saying, if here comes the rule. The rule is this to get your answer right. Differentiating these three. If you see that you have hyperammonemia, the answer must be a urea cycle enzyme. Any of these mitochondrial enzymes. All right? How do we differentiate them? Well, it should be easy. You'll see that uric acid should be high in OTC deficiency. Uric acid should be high in OTC deficiency. It should make sense. Now, why does this happen? I'll tell you why. In OTC deficiency, you have a buildup of carbamoyl phosphate. So yeah, this is where, no enzymes explain, they're not connected. These pathways are different. So yeah, let me explain it to you like this. And this should make sense. And definitely ask me questions and we'll answer it front and back. Should make perfect sense. So let's see. So what I'm saying is this, the enzyme, this enzyme here, UMP synthase, is not connected to anything here. I'm going to tell you where there is a small connection here. Yes, X-linked recessive. There's a couple of X-linked recessive diseases, and yeah, we should list them. But I'm gonna explain this. I'm gonna tell you what's happening here. Why do we have a buildup of uric acid in OTC deficiency? I'm gonna make sense of this. So this is the mitochondria right here, right? Let's say And here we have carbamoyl phosphate, carbamoyl phosphate. In order to have the next enzyme, in order to have a catalyzed reaction where you can now make citrulline, you need to have this enzyme, OTC. You need to have it. You don't have it, now we have to follow the rule. Remember what the rule was. The biochem rule was increase carbamoyl phosphate. Guess what? It increases so much because the ammonia doesn't stop. The ammonia keeps coming in. It doesn't stop. Ammonia, bicarb, and 2-ATP. This enzyme is working. The carbamoyl phosphate, one enzyme, synthase 1, it is working. So you're making more and more carbamoyl phosphate, and eventually it starts to leak. It starts to leave the mitochondria. Eventually, carbamoyl phosphate will start to leave the mitochondria. So notice this. When carbamoyl phosphate leaves the mitochondria, this is where we have the slight connection here. Not of any enzymes from the other pathway, but this is what ends up happening. You have carbamoyl phosphate from your de novo. You already have enough carbamoyl phosphate from de novo. We already had a whole bunch that we can use to make thymidine. We had that already, but there was no problem there. Now we have extra carbamoyl phosphate. Problem is, this here now becomes uric acid. You're now having so much uric acid because it's coming from two angles. It's coming from both. So, okay. Let's say you just want to get the answer right. You don't want to think too much. You don't want to be giving this so much thought. This is something very confusing to you. You can just follow the algorithm. Let's look at the algorithm. The algorithm is, if it's hyperammonemia, then it's obvious that it's a urea cycle enzyme that's not working. Yes. Now, okay, thank you. Now you're seeing that if it's a hyperammonemia, you can tell that it is a problem in the urea cycle. But you're going to be able to know what the answer is by looking for uric acid. In OTC deficiency, you'll have increased uric acid. Yeah, definitely. In OTC deficiency, you'll have increased uric acid because the carbamoyl phosphate leaks out and becomes uric acid. So you have uric acid from the de novo pathway, and you have uric acid from your urea cycle. So that's what happens. That's why you have a high level of uric acid. Does that make sense, guys? If that does not make sense, let me know. Ask me any questions. Let's make sense of this. Definitely, this is what I'm saying. Yes. Thank you. Yes, we got to make sense of this. Perfect. So what we're talking about here is that high levels of uric acid is only because we have now double the source of carbamoyl phosphate. We have one that's leaking from the mitochondria, one that was living in the cytoplasm. Perfect. We understand that. Both of them have absent citrulline because none of them can advance to the next level. I mean, look at that. They're both blocked. One is blocked here and the other one's blocked down here. Now, the other one will not have increased uric acid. Why? Because they will never make the precursor for uric acid. They're missing their main enzyme. So carbamoyl phosphate never gets made. If carbamoyl phosphate never gets made, then uric acid cannot get made, right? Okay, yes. Yeah. I mean normal or low uric acid. That's what I should have actually written there. Yeah, that's what I'm talking about, uric acid. Yeah, I should have written that. I didn't. I guess I wrote it on one side and I just said, oh, normal or low. But yeah, this is measuring uric acid. On CPS1 deficiency, you never really make it. You don't make your carbamoyl phosphate inside the mitochondria, but you cannot have increased uric acid. But okay, you guys understand that, right? And both have absent citrulline because they don't advance the urea cycle. Good, we understand that. Perfect. Super high yield. If you guys cannot understand this, you can use the algorithm that I have here. Hyperammonemia, you look for uric acid. All right, now let's say you have increased uric acid. If you have hyperammonemia, you can say it's OTC deficiency. OTC deficiency. That combination of increased uric acid means, well, increased uric acid at this stage, you're seeing this, you're saying, okay, well, what's happening here? You're not advancing the urea cycle. Therefore, you have hyperammonemia. Now, pay attention here. UMP synthase deficiency has nothing to do with the urea cycle. So the urea cycle will run properly. You will not have hyperammonemia. Look at it right here. This is a de novo pathway problem. It will have increased uric acid because of the rule that we spoke about. We spoke about the rule that if I block the, let's say, conversion of uric acid to UMP, guess what will happen? I'll have increased uric acid. Yes. So this will have increased uric acid, but it will not have a problem with ammonia. Will not have it. But you will have megaloblastic anemia. Why do I say that? Well, basically, you have an issue here making your nitrogenous bases to synthesize your RBCs, your basic cell structures. Yeah, definitely. You're having a problem here with your dihydrofolate reductase, tetrahydrofolate reductase, all that basically is not happening. So this will lead to megaloblastic anemia due to a disruption of this pathway. Yes, DNA synthesis is basically screwed. Can you get into what ways CPS1 deficiency can be called, or increase in uric acid? So let's see, hold on. So it says here, can you go into what ways CPS1 deficiency can be called? Why or what? Yeah, rewrite that question. Rewrite it so I could understand. Yeah, hopefully you guys understand. Let me know if you guys understand. Any questions, let me know. Okay, any questions, let me know, because this is important here. Definitely, you guys can just follow the algorithm. If you guys are confused, just look at it like this. It should make sense. Like they use different names or ways to describe the process, it gets confusing. Okay, so perfect. Yes, the only thing that they're slick about, they do like to use different names and different things to try to confuse you. The only thing that they're very slick about here, it's this enzyme. Instead, they just like to mention uric aciduria. You'll look for it in your books and everything, and you might not even find this enzyme. But this is the actual enzyme name that's impaired here in this step of uric acid becoming UMP. So I included it here, and I included it as well here in the diagnosis. I wrote not only the actual disease, the condition, I also wrote the enzyme. I tethered those things together just because I'm already seeing questions where the enzyme is there. That's the only thing. Other than that, yeah, they're not too slick when it comes to these questions. Now, yeah, looking at this, I can tell you that arginase deficiency is the only one here that can have a mild or no hyperammonemia for some reason. Oh yeah, look at this. This is what we were mentioning before with ammonia, making contact with the astrocytes, basically creating more glutamine. And then this glutamate coming out here and actually stimulating the postsynaptic neuron, causing asterixis, the shaking of the hand. Okay, we mentioned that as well. Those are the effects of hyperammonemia. What else we can mention? Let's see. When you consume protein, your body breaks it down into amino acids. UMP synthase is the pyrimidine synthase. So it is out of the urea cycle. It is why ammonia is normal. Yes, 100%. But your uric acid is high because that just so happens to be the enzyme right after uric acid. So uric acid is stuck. It can't turn into UMP. So therefore, it starts to build up. You see what I mean? So it is interesting if you think about it. I mean, the AB rule is followed definitely. So you're seeing that A and B, you basically cut off your A in this case is auric. Oh, I'm going to spell this wrong. Let me see. Aortic. Yeah, right now I'm hypoglycemic. Acid, let's just say. That's A. That's going to accumulate. UMP is the B here. This does not get made. UMP will not be getting made. And that's uridine monophosphate. An increase in A increase and B is not made. Yes. And this B is needed for you to actually process your DNA synthesis. So you cannot have DNA synthesis if you don't have your uridine monophosphate available. So that's what causes megaloblastic anemia. And here you're seeing that A increases, but let's understand this properly. This happens because of a deficient enzyme here, UMP synthase. Now, the other mechanism of increased auric acid was that a precursor. So let's just call this X, which was carbonyl phosphate. You had too much carbonyl phosphate coming from the cytoplasm and also coming from the mitochondria. So auric acid is increased. So you're seeing it's like a sandwich. It increases because you couldn't advance it, and it increased because you gave it too much precursor, depending on the disease. This disease here is UMP synthase. This is what they call auric aciduria, aortic aciduria. And these two different diseases can present two of them. It can be OTC deficiency, or it can be CPS1 deficiency. So how do I tell the difference? Well, we mentioned it before. With CPS1, you will not have increased auric acid because that one is before. So you don't make carbonyl phosphate. It never gets made. There you go. Auric acid increases. It has an O. So just look at the algorithm. The algorithm won't fail you. It will not fail you. You'll be able to just look at the algorithm. I'll send you guys this file. Definitely. I will put it up and you guys can just download it, or I'll see how I get this out. And yeah, just follow this algorithm right here. You guys to see if it's increased auric acid. Yeah. Okay, guys, it's 3:06. I'm already late for my next class. Yes, definitely. I'll send it to you right now on WhatsApp, actually. All right, guys. It was fun. It was fun. Thank you. I really loved it. This is actually fun for me. I actually enjoy this. This is great. It's showing the light, basically. It's like there's concepts that they just don't explain. I mean, it's like you don't find a proper explanation anywhere, and it makes it a lot easier. So definitely everything I gave today, make sure you review it, and we'll add a layer to it. We'll start to add layers to it, okay? Yeah, definitely. I would send you the notes that I have. Yeah, definitely. Oh, yeah, you're right. I was actually recording. Thank you, everybody. It was a pleasure. Definitely catch you guys probably next week. I'll see how I can fit that into my schedule, definitely. And we'll grow from what we just gave. So we're staying on biochem. We're just now going to add layers to it. Pharmacology we're going to add, and we're going to add pathology. So thank you, everybody, and goodbye. And thank you for everything. I'm going to put my Instagram. Definitely follow me. You guys can contact me. Any questions you guys have, I'm there to answer.